Welcome to the N.E.U.R.O.N conference. I'm Hans Bergman, Dean of the College of Arts and Sciences here at Quinnipiac, and we are delighted to be able to host this conference today. Our college is proud of many things, but one of the things we are most proud of is the undergraduate research we do here at Quinnipiac. And that, of course, is part of what brings you here, is your interest in undergraduate research. And the connection, I assume we will hear something about this connection, but the connection, of course, is that we believe that undergraduate research is what encourages people to, undergraduates and faculty as well, to have creative minds, to have learning minds. In other words, to build the neurons, to have the neurogenesis work is what we are excited, that's why we are excited about undergraduate research at Quinnipiac. We're proud also to have a professor in psychology, in behavioral neuroscience, who is very, very much engaged in undergraduate research and was also the organizer of this part of the conference, that is to say, breakfasts and lunch and invitations. And I'm very proud to introduce her now, and she will introduce the speaker, Adrienne Betts.
Hi, good morning. Thank you for coming and welcome to this conference, and I'd like to introduce Dr. Tracey Shors. Dr. Shors is a professor in the Department of Psychology and Center for Collaborative Neuroscience at Rutgers University. She joined that faculty in 1997. Prior to that, she received a Bachelor's of Science in both psychology and biology at the University of Alabama. She did her graduate and postdoctoral training at the University of Southern California in both physiological psychology and neurophysiology. She's been the recipient of numerous awards, including but not limited to the McAllister Fellowship, the McDonald-Pugh Program in Cognitive Neuroscience, the Pavlovian Investigator Award from the Pavlovian Society. She's fostered and mentored many undergraduate students, and that is recognized through all of her training grants with both NSF and NIMH. She has more than 100 publications, and her general research goals are to understand how memories are formed and stored in the brain. It's been recently rediscovered that in the last 10 or 15 years, the brain continues to produce new neurons throughout the lifespan. She studies the function of these new neurons in learning and memory. Her research efforts also address sex differences in the brain and how males and females differ in their ability to learn and respond to stressful experiences. Her laboratory is funded by both National Institute of Mental Health, NSF, National Alliance Science Foundation on Research for Schizophrenia and Depression. She's also very well versed in making science accessible to large audiences, as evidenced by her recent publication in Scientific American. We're very pleased to have her today, giving her talk on saving your brain, how the learning keeps new neurons alive, and please welcome her for her talk.
Okay, thank you very much for that really kind invitation and introduction. I'm happy to be here today. It's a wonderful morning, and it's nice to see so many undergraduates here bright and early and on time, even though despite daylight savings time. So I was particularly excited about coming to give this talk today because it's always a thrill to talk to undergraduates that are thinking about careers in neuroscience or pursuing professional degrees. For the most part, I do teach undergraduates at Rutgers, a lot of them, but you don't always know exactly what their future may hold. And so it's nice to see that you have taken your time out of your schedule and come on a Sunday and to hear this lecture and also to present your data, which I hope I'll get to see some of it later this afternoon. So one of the reasons I'm also, of course, interested in lecturing and talking to people about this work is because I'm so interested in learning. My background actually is in learning, more than I guess you could say neuroscience. And when I was younger as an undergraduate, I remember I took a course at University of Alabama in conditioning and learning. And unfortunately, I didn't think it was very interesting. And you know, sometimes it's the professor, but it just didn't seem interesting. You know, talking about conditioned stimuli and unconditioned stimuli and the rats and the salivation, and I don't know, I just was like, oh, not interesting. And so it's funny that, you know, so many years later, it took me so many years to kind of realize why learning is so amazing. You know, we take it for granted, right, that you can acquire information, that what I'm saying right now, somehow you've stored it in your brain so that you can recall it later. But you think, well, how does that work, right? How do you take information almost instantaneously from your environment and store it in a piece of tissue in your head so that you can recall it sometime later? So, you know, conceptually, it's a very interesting problem in neuroscience and question to answer. And also philosophically, of course, it's also an interesting question. I gave a lecture in Italy a few weeks ago to a course, kind of an advanced course in neuroscience and cellular and molecular neuroscience. So it was like PhD students primarily and postdocs. But anyway, they had lots of questions, and at the end, one of them asked me, so Professor Shors, why do we learn? And I was like, wow, that's a good question, right? Of course, I didn't have the answer. So I'm going to step back a little bit from big questions like that and talk about a relatively small part of learning. So when you think about how you would learn, at least when I used to think about it several decades ago initially, we always thought, well, you probably just change something about the neurons that already exist in your brain, right? So you probably change the strength of synapses, maybe you make more connections, maybe you tighten up those connections. But at least, you know, back in the 70s and 80s, we didn't really think that you would have new neurons. In fact, quite the opposite. You would think, well, why would you want new neurons? You wouldn't want new neurons coming in to kind of disrupt the information that was already there. Now, we believe that in part because we believe that the adult brain didn't continue to make new neurons, right? So it was easy to then explain why we wouldn't need them. But like many things you might learn, and as an undergraduate or even a graduate student or even as a professor, all things that you learn don't end up being true. So in this particular case, it turned out that the adult brain does continue to make new neurons throughout the lifespan. So, you know, this has been a pretty exciting discovery. You know, I often refer to it as a rediscovery because there were pieces of information out there, like there often are, showing that there were these new neurons. But for the most part, they were discarded, or even if someone did recognize that there were these new neurons, it would be assumed, well, there's not that many of them, it couldn't matter that much. And so a number of scientists, Elizabeth Gould, for one, who's a colleague and a friend of mine, Fred Gage, Fernando Nottebohm, of course, has always been looking at these new neurons. A number of different investigators were able to verify that indeed there were these new neurons in the adult brain. And I think that what really solidified this field was when they were actually verified to exist in the human. So this idea of adult neurogenesis has become a really popular field in neuroscience. It's really exciting, and so I'm going to show you a little bit about the work that I've been doing on these cells over the last decade. So these neurons, here's some pictures of these cells. So one of the hard things about determining whether or not there's neurons in the adult brain is verifying that they actually are neurons. And so years ago or decades ago, there weren't these sophisticated techniques, which in part explained why we didn't notice that they were there. But with the advent of sophisticated labeling techniques, confocal microscopy, internal labeling, we were able to verify that indeed these neurons do exist. So here are some of these cells. These cells are labeled with a marker, the red marker that tells you it's a new neuron, and then double labeled with something else that tells you it's a neuron. So these cells are labeled with TUJ1 or N.E.U.R.O.N, which tells you that it's a mature cell. So these cells are, you know, neurons in the adult brain that were generated fairly recently. Now, these neurons are generated throughout the lifespan. There is a quite dramatic decrease across life. So in puberty, there's many, many cells being generated, and then it tends to taper off from there. But you will generate new neurons throughout your lifespan, even into old age. So here's what these cells would look like. Within just a few hours, you might see this many cells, these green dots here. These would represent some of the new cells. And then if you followed these cells out for a few weeks or even a few months, they would start to acquire the characteristics of neurons. So they would have, I think I'm losing my, okay, here we go, here's my pointer. They would acquire the characteristics of neurons, so they would have axons, they would have dendrites, and even within several weeks they would start to generate action potentials. So for all intents and purposes, these cells, if they survive and if they differentiate into neurons, become functioning cells in the brain. Now, one of the things that's particularly interesting about these cells is many of them, indeed you could say even most of them, are generated in the hippocampus. So here's a picture of the hippocampus this is in the rat, and you can see all the different layers, cell layers of the adult hippocampus. The new neurons that are generated are in a very specific region of the hippocampus known as the dentate gyrus. So this cell layer right here where the black is, this is the dentate gyrus, and this is where the cells are generated. Now, if you look at a higher magnification, you can see these cells are very localized in the subgranular cell layer right here. And if you even go in higher magnification, you can see them labeled here in brown. So we call these, they look like little baked potatoes, actually, under the microscope. And that's because they've been labeled with a marker known as BRDU, which labels all the cells that have been generated in some specific period of time. And so one of the ways that at least that my laboratory looks at these cells is basically by counting these cells under the microscope. That's why I have so many undergraduates in my lab. Okay, so most of these cells, like I said, if they survive, will go on to become neurons. About 80%, 80 to 90% will go on to become neurons. And like I said, we'll have all the characteristic features of a mature neuron. Now, one of the things I like to always point out is that the goal of these cells may not just to become like every other neuron, right? And so they are, even though they're mature, they have mature characteristics, they're still at a different age than other neurons in your brain. So I always liken it to say, you know, all of us are adults here in this room, but clearly some of us are older than others, right? And it's the same thing with these neurons in your brain. So even though they're mature, they might have other properties that make them particularly amenable to experience, to detecting new experience. Now, these cells are amazingly responsive and receptive to their environment. So I've listed a few of the manipulations that are known to either decrease or increase the production of these cells. And it's really quite amazing. The list goes on. And in fact, almost every day you could pick up a journal and find something else that alters the production of these new cells. So the ones that have been most established, I've listed here. For example, you can increase the production of these cells if you put animals in an enriched environment. So if you put rats or mice in an environment where they have lots of toys, lots of friends, running wheels, exercise, etc., they'll produce more of these cells. Exercise is a powerful stimulator of these new cells. So if you put animals on a running wheel, rats and mice, they love to run, as opposed to lots of people. And you put them in a running wheel, they'll fight really to get into the wheel. They love it so much. And they will, in response, produce many, many more of these cells. Prozac, the antidepressant, serotonergic antidepressant, Prozac, increases the production of these new neurons. In fact, most of the serotonin drugs will increase production if treated over time. Other factors that are related to depression, electroconvulsive shock therapy, lithium, will also increase the production. So you might even be familiar that there's a kind of a pretty intense theory about neurogenesis being related to the expression of depression in humans. In any event, hormones are also powerful modifiers or modulators of neurogenesis. So estrogen, which is of course a mitotic agent, induces the production of new cells, will also increase the production of these new neurons in the hippocampus. Sexual activity, well, she was my graduate student, now she's going to be a faculty member at Ohio State, Benedetta Lunar. She just had a paper showing that sexual activity increases the production of these cells in the rat brain, in males. She didn't do females yet. And also blueberries. There's some reports that blueberries enhance the production of these cells, presumably through the antioxidant properties of these foods. So in general, manipulations that we think of as good for us tend to enhance the production of these new neurons. Now on the flip side, fewer cells are produced in response to alcohol, for example. So even relatively moderate amounts of alcohol significantly decrease the production of these new neurons. Nicotine, which is of course the active ingredient in cigarettes, decreases production. Heroin, most of the opiates, most of the drugs of abuse tend to decrease production of these cells. Aging, unfortunately, is powerful, tends to decrease. So like I said, you still produce cells throughout your lifespan, but once you reach about middle age, the numbers decrease significantly. Stress is also a powerful modulator of neurogenesis. So most stressful experiences that animals are exposed to will decrease production. So if animals are exposed, for example, to an odor of a predator, so if a rat's exposed to an odor of a fox, for example, fewer cells are produced. If you raise animals in a dominance, so that they establish dominance hierarchies, the males that are submissive, the males at the bottom of the hierarchy, they produce fewer cells than the animals that are higher in the hierarchy. Helplessness models of stress tend to decrease production of the new cells. In fact, we had a paper several years ago showing that animals that are exposed to uncontrollable stress produce fewer cells than animals that are exposed to the same amount of stress that they can control. So it's really the stress itself that seems to decrease production. Sleep deprivation, so animals that are sleep deprived produce fewer cells as well. So in general, you can see that so manipulations or experiences that we think of as good for us tend to enhance the production of these cells, and experiences that we know are not good for us tend to decrease their production. Okay, so that kind of describes their production. Well, then what are they for, right? I mean, once they're produced, why are they important, or are they even important? So one of the questions that we asked quite a long time ago now, decade or so or more, was maybe they might somehow be used in learning and memory. Now this was in part because they're so prevalent in the hippocampus. And we know that the hippocampus is necessary for some types of learning and memory, right? So if animals, including people, have their hippocampus removed, they have difficulty learning or acquiring some types of new information. They typically can remember what they knew before their hippocampus was removed, but they can't acquire new information of specific types. Now these cells, even though thousands of these new cells are made every single day, I think one of the more interesting features is that most of the cells die within just a few weeks, right? So if you label these cells and look at this population, count them if you can, and then you go back 2 or 3 weeks later, most of them are no longer there. So this illustrates this phenomena. So here's a graph of animals that were injected once with BRDU, which would label a certain population of these new cells. Most of these cells would divide, right? So now you have double that many cells in, say, 24 hours. Then a week later, you'd have even more cells. So a week after the injection, now you have 6,000 cells in response to this one injection or one label. And now, for some reason, between 1 week and 2 weeks, most of these cells die. So why? You know, it just didn't make sense to us. Why would you produce all these cells just to have most of them die? So what we wanted to know is maybe you could keep them alive by having animals learn something new. Now, most of the experiments that I'm going to talk about today, we use a learning paradigm known as classical Eiblin conditioning. And classical Eiblin conditioning is a simple Pavlovian conditioned response. I assume that most of you all have been trained in classical conditioning or conditioning and learning and are familiar with this, but I'll go over it briefly. So the animal learns that a tone, he hears a tone, is associated with a stimulation to its eyelid. And the stimulation to the eyelid causes the animal to blink, right? So the conditioned stimulus is the tone, and the unconditioned stimulus is the stimulation of the eyelid. And then the blink, which the animal has to make, is the unconditioned response. And so repeated pairings of the CS, the conditioned stimulus, and the unconditioned stimulus, the animal learns to blink in response to the conditioned stimulus. And this is probably the part where I got really bored when I was an undergraduate at the University of Alabama, because they started talking about CSs and USs all the time. But once you get the hang of it, the CS and US lingo, it's a really interesting and informative and useful paradigm. And I can go on and on about why classical conditioning is a really useful paradigm to use if you're interested in learning. But I'll refrain for now. Suffice to say that it's a really nice way to look and see whether or not an animal is able to associate two stimuli that don't occur together in time. Now, one of the things that animals learn during Eiblin conditioning is that the CS, the tone, predicts the stimulation of the eyelid and their blink. And the animal learns that within 2 or 3 trials, right? That's like a fear conditioning paradigm. The animal knows, oh, the tone predicts this stimulation. But what the animal really learns over hundreds of trials, which is what we train our animals with, is when to blink. And so the animals here, you can see, is making this eye blink response right before the unconditioned stimulus. So this blink is within 200 milliseconds, well, it's really within tens of milliseconds of the unconditioned stimulus. So this is what we would call a finely timed motor response. Now, the nice thing about it is that you can then watch as the animal acquires this response. Like I said, it'll quickly learn that the CS predicts the US. But then over many, many, many trials, it starts to really time that response exactly. And we can measure that. So it's just a really nice response to use. Now, the other nice thing about Eiblin conditioning is that a lot is known about the anatomy and the physiology of this particular learning paradigm. My mentor, my postdoctoral mentor, Richard Thompson, did a lot of this early work showing that that aspects of Eiblin conditioning are dependent on the cerebellum, other aspects are dependent on the hippocampus, and so he really developed that field of Eiblin conditioning as a model system. So we were able to take some of this knowledge that he acquired and all his people since then and adapt it to this particular question. So in one paradigm, it's known as delay conditioning. So delay conditioning, the CS, which is the tone, overlaps and co-terminates with the eyelid stimulation. And this is called delay conditioning because there's a delay between the CS and the US. But the point is that this is a really easy task to learn. I mean, animals can learn this without a hippocampus. They can learn this pretty much without most of their brain, except for their cerebellum. So it's easy. Some people call it a reflex system. Some people call it a procedural memory. Doesn't matter what we call it, right? But it's very, what we know about it is it's very easy to acquire. Now, if you put a little temporal delay between the CS and the US, so the tone comes on, and now there's a temporal gap, and now you stimulate the eye, this is called trace conditioning. And this is much more difficult to learn. Now, you wouldn't think so, right? You look and say, well, that wouldn't be that difficult. But it actually is quite difficult, because now the animal has to keep a memory trace of the CS so that it can associate it with the US later in time. Now, one of the things that's particularly interesting about trace conditioning is that it's absolutely dependent on the hippocampus. So if you take out an animal's hippocampus, they cannot learn that association. And we've done this in rats, other people have done it in mice. I mean, it's been done many, many times. And, you know, for whatever reason, if you train animals for hundreds and hundreds of trials, they cannot learn that association. So, again, it's presumed that the hippocampus is doing something to maintain this memory trace of the CS so that it can associate it later with the unconditioned stimulus. Now, the third paradigm that I've depicted here is called unpaired stimuli. And the other nice thing about classical conditioning is that then you can also use, you can present the CS, the tone, and the US, the eyelid stimulation, in a random or pseudo-random manner so that you can control for the effects of stimulation, for the fact that the animals are in this cage, that they're getting their eyelids stimulated, that they're hearing tones, right? Because animals are always learning. You know, I think sometimes in neuroscience and in psychology, well, now the animals are learning. Well, that's not exactly true. Everyone's always learning, right? We're always learning. So we're just trying to control what they're learning at a particular time. So the nice thing about the unpaired stimuli is that controls for the stimulation, learning that the CS and the US are happening. Maybe they can't tell when they're happening, but they can tell that they're happening together. Okay, so in the very first experiment we did, we injected BRDU, which will label a given population of cells that were born within a few hours. And then we wait a week so that all these cells are just about to die. And we train animals on these two learning paradigms. So one of them is the trace conditioning task, where the CS and the US are separated by this short interval of time, the one that's hippocampal dependent. And then we also train another group of animals that just receive these random CSs and USs. And then one day after they've learned, we look at, this is just the learning paradigm, showing that the animals learn. Here's the animals that receive the paired stimuli, and you can see they're increasing their conditioned responses. The animals that receive the unpaired stimuli are not showing any conditioned responses. Now, if you look at how many neurons are left in the animals that learned, you can see there's many more cells in the animals that learned versus the animals that received the unpaired stimuli. And in fact, the animals that received unpaired stimuli have no more cells than the animals, the naive controls. So the animals that were just sitting in their home cage doing nothing. Well, I won't say nothing, but sitting in their home cage. So you can see that learning greatly increases the number of cells that are left in the brain after learning. So you might say, well, does this occur in response to any type of learning? What if you trained them with other types of learning tasks? Well, if we train them with delay conditioning, that's the task that I showed you that's really easy to learn, where the CS and the US overlap in time, there's no effect whatsoever on neurogenesis. So here's the conditioned stimulus. You can see it's still on when the unconditioned stimulus comes on. The animal learns to blink, but there's no change in the number of cells that they keep in their brain. So up till now, it seems as if tasks that depend on the hippocampus are more likely to keep these cells alive than tasks that don't. So what about other tasks that depend on the hippocampus? One of the I don't know what this is, but, oh, there we go. One of the most common tasks that's used in neuroscience to test hippocampal dependence is the Morris water maze. So here's the Morris water maze. I think that probably most of you are familiar with it, but I'll review it briefly for those of you that might not be. The Morris water maze is a spatial memory task or learning task where the animal uses cues in its environment to find out where it is in space. So it would be like if you came in this room and you'd been in here before and you wanted to sit in a particular seat. And then you would come in, you say, oh, I know I sat kind of close to the clock. And then you would come back in and you'd maneuver over to your seat. Similarly, in the Morse water maze, animals are put in this pool of water, which is opaque, so they can't see underneath the surface. And there's a hidden platform. And they want to get out of the water. Rats don't really like to swim. And so they swim around looking for an escape route, and they find this platform. And they get up and they look around to see what's on the walls and, you know, what's happening outside. And then the next trial, if you put them back in this pool of water, they'll use these cues to go find the platform. So it's called a spatial learning task because animals are using spatial cues to find the escape. This task is also dependent on the hippocampus. So if you take someone's hippocampus out, a rat in particular, but humans also have some deficits in spatial learning with hippocampal lesions, they can't learn this particular procedure. So we trained animals on this particular task and looked at how many cells were left in the brain. And you could see here animals learn better over time. So in other words, their latency decreases. They find the platform faster and faster with each trial. And then if you look at how many neurons are left in the brain, the animals that had to use the spatial cues to find the platform have many, many more neurons left in their brain than the animals that could see the platform, that were left just sitting in their home cage. And we even did a stress control. So these were animals that were just put in the water with no platform and then taken out when the other animals found the platform. So it seems to be the case, at least up till now the data I've shown you, that hippocampal dependent tasks tend to rescue these cells from death more than tasks that aren't hippocampal dependent. But is this really true? So we decided to start kind of manipulating these paradigms a bit to see if it really is hippocampal dependence. I mean, you could imagine that would be a really nice story, right? So you say, well, what does the hippocampus do for learning? And then these new neurons are found there, and then you find out that they're only responsive to tasks that depend on the hippocampus. That would make a nice story. But of course, as most things go in the field, when you start to really delve a little deeper into questions, it's not always quite so clear. So in this next experiment, what we did was we took a trace conditioning task where the CS and the US are separated in time, but they're not separated by as much time. So in the traditional task, there's only a 500 millisecond trace interval between the CS and the US. And we actually shortened that to 250 milliseconds. Now, surprisingly enough, short trace, when you really shorten the interval between the CS and the US, is still hippocampal dependent. This had been reported in the literature, and I frankly just didn't believe it. And so we did it again with hippocampal lesions, and sure enough, this task is also absolutely dependent on the hippocampus. So these are animals that had their entire hippocampus removed, and through NMDA agonist, right? So it's an excitotoxic lesion, which would get rid of the new cells and the old cells. And they were trained on these two tasks, either the 500 millisecond trace, the kind of traditional trace paradigm, and the 250 millisecond. And you could see, animals without a hippocampus, there's no way. They can't learn this task. That's pretty amazing to me because a 250 millisecond trace interval is not very long. You'd imagine at some point it becomes almost imperceptible, but obviously you need the hippocampus for it. Now, what happens if you take animals with a hippocampus, with these new cells, and train them on these various tasks? Can you rescue these neurons? And you can if you use the longer trace interval task that I showed you before, but if you shorten the trace interval, so it's 250 milliseconds, there's no rescue. And in fact, again, they might as well have just stayed in their home cage with respect to these cells. So what does that mean? So that means, well, first of all, it means it doesn't matter if the task is hippocampal dependent, right? Because here's a task that is hippocampal dependent and it doesn't rescue the cells. Now, one of the things that's kind of important to realize or is maybe important for this particular question is that trace conditioning, the short trace, is more easily acquired. It's easier to learn it. So you put animals, you train them on this task, and you can see that they've already started learning a little bit faster. You know, they start off a little higher. They reach the same asymptote, but they actually acquire this task significantly faster than the longer trace. So that's when we started to think, well, maybe it's tasks that just require more effort. You know, so if you had to learn something and it takes more effort, maybe you're more likely to keep these new neurons alive. You know, and that's kind of an ambiguous term, difficulty. But we all know what it is, right? We all know when something's difficult to learn. You learn something in school, and sometimes it's really easy, and you could just, you know, it's rote. You could do it practically with your eyes shut. And then other things you really have to pay attention and concentrate. So our kind of working hypothesis right now is that tasks that are more difficult to learn are more likely to engage these cells and to keep them alive. So one way to test this is now to take the delay conditioning paradigm, which I said is pretty easy, and make it more difficult. So we did that. We took the CS, which overlaps with the US, and we made it very long. Now in delay conditioning, long is a couple seconds. So you take the CS, you extend it out almost 2 seconds, you put a US at the end of it. That's very difficult for animals to learn because they don't know when that US is going to happen, right? I mean, animals have pretty good timing systems, including ourselves, but you know, when you get down to tens of milliseconds, it's pretty hard to keep track of that. So at some point when the CS becomes long enough, it's almost like a trace paradigm, right? It's almost become part of the context if you really take it to an extreme. Because the CS is there, it's there, and in fact, when we watch the animals as they learn this task, it's almost as if they're waiting for it. You know, they start to kind of, it's almost like they're concentrating more, they're looking for it. And what they often do is blink a lot. They know the US is coming, and they just start blinking until it gets there. So it's, you know, an adaptive response. Now this task, some years ago, we did hippocampal lesions with this and found that it is also hippocampal dependent. So if you take the entire hippocampus out, animals can't learn that task very well. And interestingly enough, it does keep these new neurons alive. So, you know, here's a task. It's not a trace conditioning task, but it is difficult to learn. And once again, it keeps these cells alive. In fact, it might even be, I don't think these are significantly different, but, you know, a little more effective than the regular trace task. Okay, so the bottom line here is that not all hippocampal dependent tasks are effective, right? So the short trace, which doesn't require the hippocampus, I mean, does require the hippocampus, doesn't rescue the cells, but tasks that are difficult to learn are more likely to keep those cells alive. Okay, the next big question is it really learning? You know, a lot of times in neuroscience, we'll do a learning procedure. You know, we'll put animals in an environment, we'll train them and present them with stimuli, but that's not really learning. We're training them, we're giving them a learning experience, but are they really learning? And you'll find with rats and any species that some animals learn better than others, just like us, right? So we have lots of individual differences in the rate at which animals learn this task. So to first answer this question, is it really learning, we just wanted to block learning. And one of the easiest ways to prevent learning from happening in most tasks, at least hippocampal dependent tasks, is to block NMDA receptors, right? NMDA receptors are glutamatergic receptors. They're necessary for most types of learning. And if you block them, animals can't learn. I mean, interestingly enough, they're not necessary for memory, right? So if you block NMDA receptors, in most cases, after you've learned something, the animals remember it. So they're really, really important for learning, not necessarily for memory. So these animals were injected with a drug known as CPP. It's a competitive NMDA receptor antagonist, and then trained on that learning task of trace conditioning. And you can see they don't learn at all. Here's the animals that were injected with saline. They learned just fine, over 800 trials. But the animals injected with CPP show no learning whatsoever. And we, of course, always do these controls of unpaired stimuli just to make sure there's no nonspecific effects of the drug or of training. Now, if you look how many neurons are left, only the animals that learned, were able to learn, had more cells. So this tells you that it's not just training, right? Because everyone was trained. It's actually being able to learn this response that keeps these cells alive. The other, at least to me, more convincing data with respect to learning has to do with correlations between learning ability of an individual animal and the number of neurons that are maintained in the brain. And we've seen this in, 5, 6, probably more experiments by now. So I really feel confident about showing these data, which is, these are the conditioned responses. This happens to be between 600 and 800 trials. So you can see animals that learned well, showed lots of CRs, had more of these new neurons rescued in their brain than the animals that didn't learn well. And it's a significant correlation. This is another study. This is looking earlier in training. You can still see a correlation. So those animals that learn well versus those that don't learn have more cells in their brain. We see the same relationship with spatial learning. So animals that are trained in the Morris water maze, and those that learn well have more cells than animals that don't learn as well. Oh, and I have another example of this phenomena with looking at males and females. I know that some of you are interested in sex differences in learning, and I am too. So I thought today I would just present really just one piece of data with respect to that. So one of the things that we always find with eye blink conditioning is that females learn it much faster than males do. I should say males will learn if you train them long enough. But it takes a while. But in any event, so here's an example of this. This is the females learning really quickly right at the beginning. And here's the males kind of tagging along behind them. And interestingly enough, this is also true in humans. You can do eye blink conditioning in humans, and they find that women learn eye blink conditioning faster than men do. So if you look at how many cells remain in their hippocampus after training, you'll see that as a proportion, females retain more than males do. This is in density, of course, because the female hippocampus is smaller as a whole than the male hippocampus. But as a proportion, they rescue more cells, presumably because they learn faster, and more of them learn. Okay, so the important point is that learning must occur. So I always tell my students, you can't just show up for class, you actually have to learn. Otherwise, you might as well just stay home and watch Oprah Winfrey or something. Although you could probably learn something from her, but. Okay, so learning must occur, but it must occur with some effort. So the other aspect of this phenomena that we found is that animals that learn, so for those animals that do learn, those that take longer to do so keep more cells in their brain, right? So if you only take the animals that learn, and then you start to correlate their ability to acquire this response with the number of cells, you'll see that there's also a positive correlation. So this is actually trials to criterion. So if you take how many trials it took a particular animal to learn and correlate it, you can see there's a positive relationship. Meaning that the longer that the animal is engaged in this learning task, the more cells that are likely to be rescued. And this is another study showing the same general phenomena. Even if you kind of break it up into groups, you can see the slow learners have more cells in the end than the fast learners. The poor learners have just as many cells as animals that were trained with unpaired. So learning must occur, but it must occur with some effort. So how does this all go together then? Well, you have cells generated in the hippocampus continuously, right? They're always being generated. It's not a one-shot deal, but they're continually being generated. But if you look at one population in a day, for example, you might follow them, and 2 weeks later, most of them wouldn't even be there anymore. But if you learned, maybe almost, in fact, we did one experiment to show that if you learn well, and it takes some effort, you can retain almost all the cells that would have otherwise died. But if you just train, you just show up, but you don't learn, then the cells die. Now, importantly, most of these cells, by the time the training experience is over, they have now differentiated into neurons, right? They've become they've acquired the characteristics of a neuron. So for example, this is some neurons right after training. You can see they double label with DCX, double cortin, which is this red, and then BRDU, which tells you it's a new cell, which is the green. So right after the learning experience, these cells are now considered immature neurons, right? They have characteristics consistent with an immature neuron. If you follow them out a month later, 2 months later, they're still there. And now they're mature neurons, right? So now these are cells that these are animals that were trained. We put them back in their cage for 30 days, and you can see that the cells are still there even 30 days later, compared to the animals that received unpaired stimuli. And if you look at the characteristics of those cells at that time, they're now mature neurons, right? So they would have axons to CA3, they would be generated action potentials, and so for all intents and purposes, they would be mature neurons in your brain. Okay, so now I want to talk a little bit, up to this point I've talked about how learning affects neurogenesis, right? So now I want to kind of flip it and say, well, what does neurogenesis have to do with learning? Are they necessary? I mean, you could imagine a situation where these cells are responsive to learning, but they're not necessary for learning. So in the first experiment that we did to examine this question, we wanted to get rid of the new cells. And we used a drug called MAM. It's an antimitotic agent. So an antimitotic agent would be something that would stop cells from dividing. And in fact, most of these agents are used, for example, in chemotherapy. In fact, you've probably even heard of like chemo brain, right? This idea that when people are treated with chemotherapy for weeks and weeks on end, they often have cognitive disturbances. So we used some of these agents, these antimitotic agents, to stop the production of these new cells in the brain. So these animals were injected with MAM for 14 days, and then we trained them. We depleted the number of cells. You can see here, here's how many cells were labeled in the animals injected with saline, and here's the animals that were treated with the antimitotic agents. So they had very, very few of these new neurons, and you can see the difference here in the photomicrograph. And then we trained animals on trace conditioning. And you could see that the animals trained with, that were injected with saline, they learned just fine, right? They showed an increase in conditioned responses over trials, but the animals that were injected with MAM learned very poorly. Interestingly enough, a few of these animals did learn, you can see because the variability is high, but most of the animals did not learn. So this suggests that the cells might be even necessary for some types of learning. Now, I didn't bring all the data for these particular experiments, but I can say that they can learn most other things. They can learn delay conditioning, they can learn how to navigate in the Morse water maze, they can do context sphere conditioning. So it's not that the new neurons are necessary for all types of learning. It seems fairly specific to some tasks, and this is one of them. For those of you that are more familiar with this field, I mean, this is a very exciting, I guess you could say, and sometimes contentious question is, what are these cells really doing? What are they necessary for? And I think so far we just don't know. For whatever reason, it seems like they are doing something important for trace conditioning. Recently, we actually replicated this phenomena using another antimitotic agent known as TMZ. TMZ actually is used in chemotherapy. And so these are animals that were injected with TMZ over a course that would significantly reduce the population of new cells. And you can see once again, they just can't learn this trace response. So the animals injected with saline are fine, but those that receive this drug just can't learn. We then switched them over to delay, which is kind of an important control because you can see that the saline animals, they already know the delay condition response. They're already up here. In fact, they start to go down a little bit because they're doing so well. But the animals that were injected with the TMZ can learn delay, but it's as if they never were trained before. So for whatever reason, knocking out these cells with these antimitotic drugs is pretty devastating to learning this particular task. So what is it about learning then? What is it about learning that keeps these cells alive? Now, I wish I knew the answer to that question. We're looking, and you know, it's a really, really tough question to answer, in part because all of the key players that you would think of, right? You would think, oh, it's probably related to NMDA receptors, or it's probably related to GABA. These cells are interestingly responsive to GABA, so they depolarize in response to GABA when they're young. So it could be something related to GABA. It could be related to growth factors, you know, BDNF, NGF, you name it, IGF, right? There's so many candidates. But the problem is that almost any of those molecules or candidates that you would then manipulate would then alter learning. And if the animals don't learn, you can't look at survival, right? So it's really a tough one. But I'm going to show you a little bit of what we've found somewhat recently. So one of the other brain phenomena I've always been really interested in is brain rhythms. And we don't really know a lot about brain rhythms, right? We know they're there. We know that they change when you go to sleep. We know, you know, but we don't really know what they're there for. Again, maybe like this other question I just mentioned, they're just hard to study because they're so dynamic. They're always changing. They're not stimulus-induced. It's not like they're there or they're not there, right? Because oscillations, if you have more of one frequency, one type of oscillation, say a theta rhythm, you might have less of the gamma range. So it's a continuum. So it also makes it difficult to learn. What I would recommend, if anyone's interested really in rhythms, is to read this recent book by Buzaki. Dr. Buzaki, he's at Rutgers Newark. He wrote a book recently called Rhythms of the Brain. It's a tour de force. It's a wonderful book. I've assigned it to all my classes. I carry it around with me everywhere I go. And it's really a really great book. But in any event, so people have been interested in a particular type of rhythm known as theta rhythms for many years with respect to learning. In fact, my mentor, Dick Thompson, was one of the first to kind of associate theta rhythms with learning. And what he found in rabbits, he and Steve Berry, was that animals that had more endogenous theta rhythms, these are rhythms at about 5 to 8 hertz, learned better. And they found that when they did learn better, they produced more of these theta rhythms. Okay, so theta rhythms are rhythms that oscillate about, it depends on, again, it's a continuum, but about 5 to 8 hertz. Sometimes the range can be broader, but in any event, here's an animal with low theta, and then you can see here's an animal with high theta rhythms. So anyway, we had this hypothesis that maybe these neurons, one of the ways that they become incorporated into the brain is through these rhythms, right? So that, well, I won't go into all the details, but suffice to say that something about this rhythmic depolarization would release maybe a growth factor or something that would keep these cells alive. So recently we've been recording theta rhythms in animals that are learning or not learning, and with and without these antimitotics. And somewhat surprisingly, although I guess that's what we proposed, still surprised to see it happen, is that the animals that were injected with the TMZ, the antimitotic agent, produced less theta rhythms than the animals that were injected with saline. Now this is before any training happens, right? So in other words, if you take out these cells, what you think about it, you know, there's not that many cells. I mean, it seems like a lot when you say, oh, there's thousands every day, but compared to how many neurons are in the dentate gyrus, it's not that many cells. But for whatever reason, you take these cells out, and now there's these theta rhythms are disrupted. And again, you know, it's hard to say what the consequences of that are. I mean, you interrupt one rhythm, and that's going to change most other rhythms in that, at least in that region. And that may actually then explain, I'll use that word cautiously, but may suggest, let's say, why they can't learn, because now they don't have these rhythms. And this just shows the data a little bit more clearly. Here's spontaneous state activity before training, right? Before they receive the CS. This is some before, not in response to the CS. This is during training. And you can see the animals with the saline produce more than the animals with the TMZ, even once training begins. This correlates, of course, with the learning. So anyway, this is just one of the ways that we've been thinking about how these neurons will eventually become part of your brain. Right? They're not operating in isolation. They're not isolated units, but they're connecting up to all these other neurons, which then, in turn, presumably have some impact on the way the animal learns in the future. So this is kind of just a little diagram. Here's the old cells firing. Here's some of the new cells that are immature. Maybe some of them are dying, but then they get recruited with these rhythms in the CS, induced rhythms, to survive, and then eventually all of these cells are able to fire in synchrony. Again, this is kind of our working hypothesis at the moment. Okay, to just kind of summarize, I guess I'd like to put this in a little bit bigger context just for a few minutes. I think I have a few minutes left, I hope. So I think in neuroscience, oftentimes we think about things unidirectional, in a unidirectional manner. So we'll say, well, learning affects neurogenesis, and neurogenesis affects learning. And that's great, you know, that's how we design our experiments, but, you know, in the brain, it's probably some kind of elaborate feedback system. And I'd like to think of this, the relationship between learning and neurogenesis as a positive feedback system that keeps your brain healthy and fit for learning in the future. Right, because a lot of times also I think we think, oh, the purpose of learning is to store a memory. Well, not exactly. I mean, memories are useful. They're even fun sometimes to like reminisce and think about. But I mean, the reason you record something and remember it is so you can learn better in the future, right? So really learning is for learning in the future, preparing yourself so that you can learn more rapidly. So I like to think of it as that the fact that new neurons are produced, these neurons are kept alive by learning, and now you're able to learn better, right? You have a more efficient, integrated brain with these new plastic elements that allows you then to keep more new neurons, and it becomes this kind of positive feedback system. Okay, so at some point you might say, well, okay, but these are rats, right? So what about us? It's not like all of us are going to go home and set up an eye blink conditioning paradigm in our kitchen. Well, some of us might, but most of us probably wouldn't be so inclined. And so we've been trying to think, like, how could we broaden this, right, and make it more applicable to people and how they can think about their own brains. And, oh, I put this up here because sometimes, one time when I gave a talk not too long ago, someone asked me about reading. I said, well, what about reading? And I said, well, I don't know, because rats don't read. And so I just couldn't really answer it. So I started thinking, well, you know, I'm sure it depends on what you read, I would imagine, but is there anything else that we could use in a rodent species that might have some analogy to humans? And so we recently started using what I call it a physical learning paradigm. The people in my lab actually call it their version of jazzercise. But it's a training procedure where the rats are on this rotorod, which I'm sure most people here are familiar with. It's a rod that turns at various speeds. And they use it in pharmaceutical industries to test to see whether or not animals have motor deficits in response to drugs and what have you. But we took this rod and kind of developed it and made it more like an exercise task. So the animal gets on the rod and they like it generally until it starts going really fast, right? So then the rod goes faster and faster and faster. And some rats really love it. You know, they're like gym rats, right? They love it. And then other rats are like not so good at it. And so if you train them long enough, they do eventually acquire this task. Now, interestingly enough, we don't even think this task is dependent on the hippocampus, because we did hippocampal lesions and they still seem to be able to do it. But it does seem to keep these new neurons alive. so we inject the animals with BRDU, right? So these are cells that are already there when the animals start the training. A week later, they're trained on this task, and you can see they have more cells than the naive controls. We also just put animals in an exercise wheel, so they didn't have to learn anything, they were just running. And you can see there was really no effect on cell survival. Remember, these are survival, not proliferation. And it does keep some percentage of these cells alive. Now you can see this effect is not nearly as big as the effect with trace Ivalent conditioning or as in magnitude, but still you're keeping cells alive that would otherwise have died simply by engaging in an exercise activity that requires learning, right? It requires attention, focus, and perseverance. Oh yeah, I just wanted to show, if you take the animals that were able to learn this task versus those that didn't really do so well, then the effect is even more pronounced. So again, it's a learning phenomenon. It's not just being exposed to this procedure, but it's rather learning it and mastering it. So, how can we save our brains? Well, first of all, you have to make these new neurons, right? And you have to have something to work with. And so we do know of various ways that we can enhance the likelihood that these cells will be produced. Exercise, like I said, just regular old exercise, aerobic exercise is a powerful, will increase the production of these cells. Antidepressants, I'm certainly not advocating that, but I mean, in some cases, I'm not. But you know, they do increase production, good nutrition, adequate sleep, sexual activity, friendship. I mean, all these things that we know are good for us likely increase the number of these cells. Whoops. And then you have to keep them alive somehow. And you know, we can all think of ways to do that. You all showing up here on a Sunday morning, that's probably a pretty good start. I try to think of things that are really mentally taxing, right? So problem solving, obviously, scholarship, psychoanalysis. I've actually learned a little bit about psychoanalysis recently, and it's pretty taxing if you really get into it. Some types of sports, obviously, are mentally engaging, and music, playing music, I didn't even think of that, put that up here, symphonic music. Cognitive behavioral therapy, as most of you know, is pretty difficult to do. That's why people would rather just take some medications than engage in it, because it's difficult. Mindfulness, which is kind of a new wave in psychology, right, where you learn to recognize the thoughts in your mind and maybe even learn to control them to some extent. Zazen, which is a form of meditation, which is really difficult to do for those of you who have ever tried it, because you're trying to think of nothing. Dance, I would imagine, is probably useful, and of course science, which is always engaging. So anyway, in conclusion, I wanted to read this letter that I received from an elderly man some time ago, and I thought he kind of encapsulated these ideas. He said, "I have come into neurogenesis and the activity of the hippocampus indirectly. My first concern is the fact that education seems to prevent Alzheimer's disease at a later date." That may or may not be true, but I thought this was interesting what he said, is, "I wonder if I may continue thinking as a result of your remarks and go on to say that Alzheimer's will occur more often in the senior years if there has not been learning during the formal years to save the original neurons in the hippocampus." So since this is a nice large group of undergraduates and apparently even some high school students, I think that's always good advice, right? Learn as much as you can, live well, be curious, and always keep learning. Thank you. I also really want to acknowledge all the people in my lab who have really done an immense amount of work counting all these cells, conducting these experiments, being enthusiastic. And I especially, again, for this conference, want to thank all my undergraduates. I have quite a nice group right now, and they've been wonderfully inspiring for me. So thank you. I'll take some questions if anybody has any.
that the cells continue to survive? Once they mature into new neurons, do they survive indefinitely? And what is the cutoff for cells surviving for like a long period of time? Is it when they mature into new neurons, or would it be sometime before that?
Well, you know, it's difficult to say. We only look 2 months. You know, it's hard to get students to look beyond 2 months. But you know, intuitively, it seems to me that once they're rescued and once they reach that 2-month period, they're probably there indefinitely. And that's just data from my lab. Other labs show that once the cells become incorporated, they're probably there indefinitely.
I was curious about once these cells get stimulated by the attention and making this association, then you immediately could implement an extinction paradigm and make the beneficial effect on neural retention go away.
We've tried to address this extinction issue. And I don't think we've quite done that. I mean, of course, many would argue that extinction is just new learning, right? That you're not really extinguishing. You're extinguishing the response, but you're really learning something new. And there have been a few studies suggesting that these cells also might be responsive in a positive way to extinction. So, no.
Do you think that cell death is balancing the rescued numbers, or that you're packing more in?
Yeah, you know, I didn't bring the data for that particular question, but I am often asked, you know, what would happen if you just learned and learned and learned? You know, would your brain eventually just explode? And I said, well, I've never seen anybody's brain explode, but we do have some data suggesting that the cells that are slightly younger than the 1 week of age cells are less likely to survive. So the cells that were around 30 minutes, which of course is, you know, within hours of being born, when the training starts, are actually less likely to survive. So there may be some kind of feedback system where when the one group population is being rescued, the next group is not so likely to survive. And others have found similar effects. N.E.U.R.O.N has some similar data with spatial learning.
What about killing off the existing population that's already there? So the older, I would have expected that the older cells would be culled in preparation for recruiting a new cohort.
I don't think there's any data to support that. And as you know, it's really difficult to do those studies because you'd have to be there right at the right time to catch them when they die. So I don't think that's known.
Mm-hmm. So you said that you saw that the cells that the cells that degraded if they weren't used for learning would die off, right?
Right.
So what if you use a nerve protection and that prevent them from degrading, prevent the brain from keeping on making new cells?
Right.
For the ones that were used.
I mean, so if you could inject something and keep them alive so they don't die, yeah, that's a possibility. And there are certainly a number of studies suggesting that there's ways to keep the cells from dying.
Right, I know what you're mentioning about Alzheimer's. Now that you use Alzheimer's research, so.
Oh, yeah, that's a good idea. I think there's people working on that now, but we actually don't have any data in that regard. We are really interested in looking at nerve growth factor for kind of reasons related to that. But yeah, that would be great.
I was just wondering if you've done any research on how caffeine affects.
How caffeine? I haven't done any research on that, and I guess I don't want to say actually what I think it. Does anyone know? I don't know if it decreases or it increases it, so I don't know.
Related to that question, I was sort of thinking along the same lines. The things on your bad, things that decrease cell survival. But there is evidence that, at least in low doses, psychostimulants like nicotine, caffeine will enhance attention and enhance learning.
Right.
Sort of rectify it.
Well, it's a little bit different situation. So the list that I put up, that was for cell production. Right, so that would be for generating more cells as opposed to keeping the cells that are already there, right? And so learning keeps the cells alive that were already made, but if they were never made in the first place, there's nothing to keep them alive, right? There's nobody to keep alive. It's a kind of a subtle distinction, but it's an important one, right? So you have these cells, and you either produce them, and if you never produce them, there's nothing to keep to rescue. So you have to produce them in the first place. And then the survival effect occurs after they're already there, right? After they're already sitting around. I hate to say sitting around, but after they're already around for a week, that's when we stimulate them. Now, obviously, if you make a lot more cells, so say you went on a big exercise routine and you produce many, many more cells, so, more would tend to survive, right? Because some proportion always stay alive. But that's a different type of survival than actually rescuing cells that would have otherwise died.
Do you think there's a difference in the cells that are susceptible to experience during this time period and the ones and die off, and then that sort of, it's a relatively large portion that remain. Do you know what I mean? As it doesn't go back down to zero. So do you think there's actually two cell types? The one that's always continually being produced, that's regenerating the hippocampus, and then another kind that is susceptible, or do you just think that you're seeing a decrease in the proportion of the same type of neuron?
Right. That's a great question, and I don't know the answer. I mean, again, it kind of gets back to this issue or this point that all animals are always learning, right? So we can't ever have an animal that's not learning. So it could be that the cells that are hanging around are the ones that were rescued by something that we don't really understand. I mean, that's why, even these experiments where they use enrichment, there's sometimes it's difficult to decipher if those effects are due to cell production or survival, because in an enriched environment, there's so many learning opportunities, and you don't always know when they're happening. So I don't know the answer. Yeah.
Can you examine the structure of mature neurons, so in relation to spine density or synaptic connections?
In response to learning, or just in general?
New mature neurons.
No, I haven't. That would be great to do. That would be really great to do.
When you said before about taking longer to learn, seems to keep the self alive. Kind of like a quality versus quantity, do you think spending more time, period, or the difficulty of the study?
Yeah, that's another great question. I guess my intuition says that it's the time. They're kind of related, of course, but I haven't thought of a good way to dissociate those exactly. There probably is a way to do that, but it seems to me kind of intuitively that if you take yourself kind of out of the context and you think about just the cells in the hippocampus, that the longer that they're firing and being exposed to these rhythms and firing during the training experience, then the more likely they're to release some growth factor or whatever that's eventually keeping them alive. That would be my intuition. Yeah. Okay, I guess that's it. Thank you so much.